Hello everybody, I am Dr. Marzoog Basheer, a PhD student at First Moscow Medical State University, and I have published a paper in the journal Cardiovascular and Hematological Disorders in Drug Targets. The paper is titled Autophagy Behavior in Post-myocardial Infarction.
So we can see here the paper Autophagy behavior in post-myocardial infarction, which is aimed to determine the change in the autophagy in the patients who survived a myocardial infarction. We can see here the abstract. So we can see the mechanism, how does autophagy affect on the injured myocardial. We see that injured myocardial, they suffer from the ischemia reperfusion injury, which has primarily three effects on the JNK pathway, the mTOR-C2 pathway, the JAK2 STAT3 signaling pathway. We see that JNK pathway, it either induces apoptosis or autophagy, depending on the degree of the injury to the myocardial sites. If it is severe enough, it induces apoptosis, the death of the cell. It is the program type of death. If it is not very high level of injury to the myocardial sites, it induces autophagy, which is the best choice where there is rescuing for the injured myocardial sites. This pathway autophagy can be, we said, through mTOR-C2 can also induce autophagy, or the JAK2 STAT3 can induce autophagy at the same time. Maybe three pathways are at the same time. Maybe separate one of these pathways induces autophagy. The next step, it is the pickline one pathway. It is the induces the or upregulate the ATG8 and the RC3 too, which is responsible for the formation of the autophagosome. The JAK2 STAT3, it can be also inducing ATG8 and RC32 formation through the RC31, not only through the peak line, yes, peak line 1, but through also the RC31. The autophagosome, it induced lysosome function, which is like a vicious circle. That lysosome will also result in the autophagosome formation. We see the final effects of the autophagy on the injured myocardial site. It is a reduction in the reactive oxygen species formation, which is the basis in the pathogenesis of the damage to the myocardial site, damaging myocardial sites, and the decreasing in the energy expenditure, and the reduction in the endothelial reticulum stress and lactic acid formation, which is typically seen in injured myocardial sites. All these cumulatively, these effects, they result into the dormant status, the myocardial site pass into dormant status, reduction in its contraction, proliferation, and so on. Nevertheless, despite that there is a low level of the proliferation, but it is present. So we can see the effect of the autophagy is period dependent. So according to the results of our study, that autophagy has a protective effect during the early period of myocardial infarction or injury to the myocardial sites. However, on the other side, if we induce autophagy in the late period, it usually induces further damage to the already injured myocardial sites. Therefore, probably induction of autophagy is more protective, has more therapeutic effects in the early period, which is difficult to say the exact time for the early period. Usually, probably, as we suggest, it is less than uh, 24 hours, or in the early period, in the acute period of the MI, less than 7 days. Also, there are some agents, some moleculars that are used for the treatment of other diseases, and some of them they are under investigations for the treatment of autophagy uh, disbalance in the uh, lung fibrosis, such as resveratrol, and some drugs they are antisenescent agents, such as sirtuins and also the NADH, and it is byproducts. So, in the finally, we can say that autophagy has a therapeutic effect in the early period of myocardial infarction and more damaging effect in the late period. And in each period, there is a different signaling pathway they are activated, and each signaling pathway are potential targets for future therapeutic strategy according to the period. So, if the patient or these myocardial suffered from myocardial infarction uh, from early time, we can induce or elevate these signaling pathways or these uh, molecules, yes. But if the patient or these myocardial have suffered from ischemia or for a long time, we may inhibit this signaling pathway or induce this signaling pathway. So in the final results, we can get a protective effect. Thank you.